Welcome everyone to the European Coral Reef Symposium. We are going to opening this conference. Before of all, I would like to thank all of you to coming, and my co-chair Carlo Serrano. I want to tell you that this adventure started a couple of years ago. Actually, we involved many colleagues and friends, starting from the Italian colleagues that participate in the local organizing committee. Most of them are present today here, so I want to thank all of them. Then, together with them, we established a scientific committee for this conference. All of them, of course, are present today in this room. I really acknowledge all of them for the contribution they provide in the organization. I will also tell you that we have a Green Strategy Advisory Board, because one of the commitments of the conference is to be at least neutral in terms of carbon footprint. We will tell you something about that later and in the conclusion of the conference. And we also have a student and early career advisory board. We provide for some award at the end of the conference. So, Carlo?
Yes, thank you all for coming here. And I think we can start with the official presentation. And we have, of course, to thank a lot the Stazione Zoologica of Naples for this, and Professor Antonio Terlizzi is here to represent the station and will bring some presentation of the station.
Thank you, Carlo. Thank you, Massimo. And good morning, everyone. And I am more than proud to be here today for a number of reasons. First of all, because I represent here the president of the Stazione Zoologica, Roberto Bassi, who was impossible for him to stay here together and ask me to introduce and to make you his salutation. I think that most of you know the Stazione Zoologica. It is one of the most prestigious and ancient research centers all over the world. And I already said that I'm proud to be here, because not only because the ECRS Congress represents most of my research activity, but also because it is a big Congress. And Carlo said to me that you are about 700 people from about 60 countries all over the world. It is a very important Congress, and it makes me proud also because I am a Napolitan. Napoli is my city, and I'm very glad to see a lot of science who belongs to my research activity here in Napoli. So thank you very much on behalf of the Stazione Zoologica to be here. Thanks to all the organizers. Please allow me to thank most my friends and colleagues, Carlo and Massimo, and all the organizing committee to be here. Enjoy the Congress, have a good work, enjoy Napoli, and enjoy the food, the city, the art, and if you have time to do this during your work. Thank you very much, and welcome in Napoli.
Yes, now just a few words from Gert. Okay.
I like to stand up when I speak. Good morning, everyone. Fantastic to see you all here. Glad we all made it to Napoli. On behalf of the European chapter of the ICRS, I would like to warmly welcome you to this European meeting. First off, big thanks to Massimo and Carlo and everyone involved in their team in the local organizing committee to put all this together. That fantastic event that brought us all here during the following for the next days. The European chapter of the ICRS is recent. It was formed in 2019, just before the COVID pandemic hit. And we have some main aims and objectives to pursue the mission of the ICRS in Europe, of course, and to organize and support holding of European conferences. And this is what we're here for. Until now, these meetings have been organized more or less ad hoc, just by voluntary work by colleagues. Apparently, Rupert Ormond told me that they already started in the 70s and 80s, but he couldn't recall where it was. In this millennium, they were held in 2006 in Bremen, organized by the Bremen crowd, by Georg Heiss and his colleagues. In 2010 in Wageningen by Ronald Orsinger and Detmar Sipkema. Georg I have seen here, but the two colleagues from the Netherlands I haven't seen here. 2017 in Oxford by Mike Sweet and colleagues, and now here, thankfully, in Naples. Ideally, the European chapter would like to have them in 4-year intervals spaced between the international symposia, and we see here if we can achieve that in the future. The chapter, as you have heard, has been involved in the organization of this conference in various ways. For example, serving on the scientific committee and supporting the organizers to make this an official ICRS event, and we're very, very, very happy about this. European science continues, European scientists continue to be at the forefront of coral reef research globally. Although we don't have shallow water coral reefs at our doorsteps, we do have overseas territories, we do have deep water coral reefs, for example, along the Norwegian coast. So the theme of this symposium, Bridging Knowledge Gaps Between Tropical, Temperate and Cold Water Coral Reefs, is timely and well conceived, and I'm sure we can all contribute. And who knows what the future will bring. It doesn't look too good if we look at the news that the fossil fuel consumption worldwide has peaked this year. It's a little bit depressing. But on the other hand, me being a geoscientist, the future is always different than the present, and maybe we see coral reefs in the Mediterranean in the not too distant future. Of course, this would be interesting, of course. And the current very exciting scientific program of this ICRS is testimony to the forefront where the European research in coral reefs is playing. And it's very encouraging and very good to see that we have so many young people here, so many graduate students. That's really, really fantastic. I wish you all a very successful symposium full of exciting science and fruitful discussions, meeting old friends, making new ones, and working towards a better science-based solutions for the keeping of the coral reefs that we all love. Let me finish with an invitation to our chapter meeting, which we will hold at 5 o'clock tomorrow in Archimedes, I think, where we will give more insights into the chapter and hopefully decide on the next European Coral Reef Symposium. So if you're interested to host a wonderful event like this, let us know. Thanks.
Okay. Please, Christian, President of the International Coral Reef Symposium. Please.
Thank you. Good morning, everyone. Gerd already said most of the important things. I just want to remind you, use this conference also to get to know people, and for the students, don't be shy to talk to the professors. I remember when I was a student, and I was super intimidated, and there's no reason. People are usually pretty friendly, as it will turn out. So use this to collaborate or to establish collaborations, start to think about new projects and so on. I also want to remind you that ECRS and ICRS is a voluntary organization, and if you feel compelled to make an even further difference, feel free to reach out and think about if you want to take a spot in the society, a more active spot. With that, I would conclude with let the science begin and the research. Thank you, and I see you around.
So thank you, everybody. Just a few technical notes to starting well. As I think most of you already knows, we launched a questionnaire to collect information of your travel accommodation here. That will help a lot to correctly calculate our footprint. So if someone still not please fill the questionnaire. You can also find the QR code outside and on the website, of course. You know, we have 13 very exciting sessions, 5 parallel sessions every day. So I will acknowledge all the chair that already did a very big work, and they are going to a very hard task in these 3 days. To follow the presentation, you have all the resources on the website, and also a fantastic smartphone app you can download. It contains everything, including the abstract of the presenting people and the poster. So please use this. And you also have on the website the perfect time. I will ask all the people that are going to present the oral speech to pay attention to the time. It is extremely important to allow people eventually to exchange room for attending different sessions. You know, at the end of the conference, all of you are invited to submit your research outcome for a possibly publication after, of course, a peer review process on the Coral Reef Journal. That is the official journal of the ICRS. I don't know if Eva is here in the room. Okay. She is from the publishing editor of Springer. She also have a stand, a booth of side, and please refer to her for every information. Thank you, Eva, to come. Okay, and just to finish, I want to thank everybody. So we may start with the plenary session and the plenary speaker. Thank you so much.
So I have the honor now to introduce the first plenary speaker of this symposium, Dr. Marta Ribes. She received her PhD in biology from the University in Barcelona in 1998. The topic was on benthic feeders metabolism using in situ incubation chambers for Gorgonian species, sponges, and ascidians. And I think that broad theme has followed her until today. She then did a postdoc at the Hawaii Institute of Marine Biology, working on the significance of picoplankton in coral reefs and the role of hydrodynamics in nutrient acquisition. And in 2001, she returned to Spain and received the prestigious Ramon Nicayal Contract Fellowship. And since 2007, she's permanent staff at the Institute of Marine Science in Barcelona, Spain. And her research is mainly on coastal marine benthic filter feeders on rocky substrates, with focus on energy dynamics and metabolism of the dominant taxa. Welcome, Marta. We look forward to your talk.
Hello, everyone. Dear organizers, Carlo, Massimo, dear and all the committee, dear participants in the 2024 European Coral Reef Symposium, it is an honor for me to be here today. I was not expecting this. It is a busy time for us in our little corner of the world, but you know, I didn't want to miss the possibility of giving a talk in front of this, you know, outstanding audience, also in this magnificent place, visiting Naples also, and mostly, you know, finding so many colleagues here and spending time with friends. So I thought, let's go for it. So thank you very much for this opportunity. I would like to start my particular journey. Let's see if this is working. No. With a heartfelt thank you to all my collaborators, co-authors, and you know, all of them are a constant source of inspiration for me. This is you know, team journey. So all what I will say is not just me, is that us in a broad sense of the word. I think the maintenance of life on Earth depends on the effective functioning of the ecosystems. Among them, we have tropical, temperate, and mesophotic water reefs. All of them are topic of this symposium. And you know, all of them host a high biodiversity and also unique ecological processes. These processes provide us on many goods, which are known as ecosystem services, which include food, protection, materials, climate regulation, transport route, recreational areas, and cultural benefits. However, we all know that we are at the Anthropocene epoch, and these ecosystems are being affected by, you know, and the humans are recognized as a major force of change at the planetary level. So all these services that this ecosystem provides are being endangered, right? So these human-induced stresses are generally known as the general name as global change, but include habitat destruction, climate change, invasive species, overexploitation, eutrophication, and other types of pollution. So all these stresses are disrupting the functioning of the ecosystems and are modifying the biomass, so they are putting in danger all the goods we are getting from these ecosystems. This inspired a bunch of books telling us, explaining us why do we need the nature, why we need the wild, right? And also inspired worldwide initiatives, you know, and with the organization of different nations going to fight for this common problem and proposing ambitious goals, I think, such as the 30-30. The 30-30 is the protection of the 30% of the ecosystem by the year 2030. It also comes along with, you know, with a 30% of restoration of the degraded ecosystems and, you know, reduce the food waste, watch out the algae species, and also the excess of nutrients, between others. So, but the implementation of all these ambitious goals need to be rooted in a deep understanding on the ecosystem functioning. Function-based studies have opened a new, you know, a new window for us in our way of approaching the ecosystems. But this term is not free of controversy. In fact, I mean, we use it a lot. We say functional diversity, functional identity, functional redundancy, functional ecology, functional classification, and more, a lot of them. But the main thing is to go from the measures of functional traits to upper, you know, organization levels and ending to understand the ecosystem functioning. And as associate to that, we will know, I mean, the ecosystem function, and if those functions are good for humans, we call them services. However, some authors, you know, make a revision of the term function and say that we have the tendency of measure things that we know how to measure and then functionalize them, you know? So, and this is very good and collaboration. So they propose to do the other way around. So look for a particular function and then look for the traits that will be good to measure, you know? In this sense, these authors propose the use of the movement or storage of energy and material. And doing that, they say that, we will be able to manage and to understand high diversity systems such as the coral reefs. This is pretty much the way I saw this ecosystem functioning, how I approach to the ecosystem functioning. So going from organisms' functional traits and scale it up at the different levels of the organization. So in this sense, to this end, we use what we call the scope for growth. The scope for growth is the difference between the energy acquisition through the feeding and less the energy for maintenance, so the cost that it takes an organism to maintain the actual biomass. So this difference gives you like an amount of energy that you can invest either in secondary production, either growth, reproduction, or defense. Right? So by using this approach, this energetic approach, we wanted to measure these traits, feeding rates and respiration rates, under reliable condition, that means in situ, right? So during my PhD, we designed a prototype where we were able to estimate the feeding rates of different benthic invertebrates, and also in the same system, and also including all the potential foods, because the traditional analysis of gut contents were not including the small particles such picoplankton because the size and because they don't leave any remains. So, and also this system allows us to estimate respiration rates by means of a polarographic type sensor and measuring the oxygen concentration inside the chambers, the experimental chamber with an invertebrate and the empty chamber as a control, and we were following, the decrease of this oxygen through time, and then through calculating these slopes, we got the respiration rates. By using this, by measuring these traits along the year in 3 different Mediterranean species, we got to explain the seasonality observed in some of these groups. So as here you have all the months of the years and different groups of Mediterranean benthic invertebrates, and what you can see, and the colored lines are just secondary production or activity. So what you can see marked in red is that during summer, July to September, was like an empty space in terms of investment, meaning that there was, we suspected that, this was because some energetic limitation. Also, when there was something, this red line is an active inactivity situation for some of them, for example, as Alcyonium, that they cover themselves with a white, thin layer, so they are inactive during this period, right? So we suspected then that, this can be related with an energetic, they were deprived of energy, right? So what we did then was calculate the scope for growth for 3 species, 1 Gorgonian, 1 Spongian, 1 Ascidian, and what we saw that this is the scope for growth. 1 means that you are able to keep your biomass, not do anything more, and more than 1 means that you can invest, growing or whatever. So what we saw was that for the 3 species, summer was the more limited season around the year, right? And the summer conditions in the Mediterranean are characterized for having, there is a strong stratification, so there is few food available for these shallow communities, and also it has the higher temperatures along the year, right? So then what we tried to do with the Gorgonia Paramuricea clavata was to experimentally, elongate this period and in the lab to see if we could explain the mass mortality events that we saw in the Mediterranean of this species, you know. So we were able, what we did was just to elongate. It was not a thermotolerant experiment, was just keeping the highest temperature, but a normal temperature, not a lethal temperature, and then just deprive them of food as they used to be in summer, and we elongate there for more time, not just the time that the duration of the summer. So they started to get thinner and thinner, and in the end, they started to die. So meaning that these mass mortality events, were the trigger mechanism was this energetic, an energetic process. So we were able to explain, this ecosystem type of thing. So, and then we tried with this, measuring these traits, we always try to, to combine different methods and when needed to improve them. So for feeding, and we try to combine, chambers with the INEX method, which allow us, and depending on the question, it allows you to measure at the same time the inhale water and exhale water directly to the, the vials that go directly to the analysis. So, it's a complementary method for studying feeding rates. I'm sorry, I'm not sure I know what is the sensor. And here, thank you. So then for respiration, we also change. We first change from the polarographic sensor to this, optical sensor. At the beginning, we want, and again, depending on the question, during Teresa Morganti PhD, what we wanted to do is to have a snapshot of the respiration. So what we did was measuring, the inhale water and exhale water in these tubes where the spot sensor were, and we measured then the oxygen concentration. So the difference was the respiration rate. But now we are using a much fancy system with optodes, and what allow us to have, measurements of oxygen from inhale and exhale water through every 10 seconds or few seconds for a couple of days. So this is the type of data we have. So you have temperature and you have, the ambient water and also the oscular water, oxygen concentration. So it allows us to integrate, the variability through a longer time period. So in this case, it's an improvement, it improves our capacity of measuring respiration rate. So at some point, all of this is organism level studies. No? So at some point, I feel that, okay, but those organisms are living with others, they have neighbors. So what are the effects on having neighbors, and also what is the effect of hydrodynamics? So to find this out, I moved to the Hawaii Institute of Marine Biology in Kaneohe Bay, Oahu, and by using a flume system, we studied we wanted to know if the nitrogen uptake by the two most common communities there, which are live coral and rubble, which is dead coral reefs covered by turf algae, were able to remove picoplankton. Until then, most of the studies of nutrients removal was done with inorganic sources, so ammonium, and they found that when they measure uptake rate for the both communities along different water velocities, they found both communities follow the same pattern, meaning that no matter it was coral, live coral, or was turf algae. So meaning that the control of this uptake was a physical control. They call it mass transfer limitation, right? And this is that the limitation comes for the transport, or in this case ammonia, through this depleted boundary layer near the phototrophic surface. So, and this transport, was the limitation step, right? When we did these same experiments, what we saw was a different pattern. So when we calculate the picoplankton removal rates by the same communities and also the same water velocity range, what we saw was that live coral was both communities were removing picoplankton. Rubble was removing much more than live coral, and rubble had a positive effect of the water velocity. So we suspected there that this was not due to corals then, because rubble, is dead coral. So we suspected about the associated fauna living, inside the corals or the rubbles. So what we did then was we manipulate the amount of these filter feeders by, exchanging rubbles with the skeletons and also rubble with the new live coral, meaning that we took the live coral heads and we removed sponges and ascidians, and what we got was the same, the same relationship between uptake and for both communities and the amount of either sponges or number of ascidians. So in this case, the control was a biological control, and the difference between them, between rubble and live here is just because rubble has much more, associated fauna. Okay, and the position inside the head is different. So we find out that this and was a different control, okay? So then we also find out that the nitrogen from picoplankton was more than 90% of the nitrogen of removal for these communities. This was published in a couple of papers back then, and I would like just to make an insist of a brief memory of two great scientists I had the honor to collaborate with, the zoologist Robert Kinzel III, Dr. Bob, and also the biogeochemist Marlene Atkinson. So then back to the Mediterranean and continuing with these community studies, what we wanted to know is in the coralligenous community, which was the effect of the presence of the gorgonians, the physical, the structure, in the removal of picoplankton by the sponges living, cohabitating the coralligenous. So to this end, we built a flume canal in my present institute, and what we did, so the first was vertical velocity profiles. This is the flume depth, and those are different water velocities. So the first thing we saw, the empty things and the empty spots are the profiles with gorgonian. So what we saw was an acceleration upper the gorgonian, then translated to shear stress, which is a measure of, let's say, turbulence, okay? So represented a decrease of turbulence just at the level where the sponges were pumping. Sponges doesn't like much turbulence. So this, wider the range of hydrodynamic conditions where the sponges were capable of removing efficiently the picoplankton. So this was a nice example, of facilitation between species living in the same ecosystem. Okay, until now, I talk about, energy and material fluxes and considering as a functional trait, so individual nutrient fluxes, individual oxygen fluxes, also community nutrient fluxes, and a little bit of the hydrodynamics, and how all these measurements help us to explain seasonality, distribution of secondary production, even mortality effects, or the physical or biological control. But I must say there was by the beginning of the 2000s, there was like an inflection point somehow that it changed our mind in the way we were approaching to the ecosystem. And this is just because we were measuring, fluxes in sponges, as some of the species were excreting nitrate, and that was like nitrate. No, they should excrete ammonia, right? And then we started, so yeah, they are not alone. They are, they live in a community, right? So this switch to looking at the organisms as holobions, definitely it marked, our way of approaching the ecosystem. And meanwhile, I was, preparing this talk, it came to my mind all the Gaia theory and the philosophical ecology, right? And exposed by Lovelock and co-developed by Leon Margulis. So, I mean, it's like this looking at the Earth as this massive self-regulation organism, right? But more than that, what I like is the holistic view they had, so the need of considering all this interconnection between the system, the living system, and of course also including the humans. So later on, I mean, now we have clear idea that the microbial participation in the fitness of the holobiome, right? So it's not just a nutrition thing, it's also a protection, a development, behavior, detoxification, and also temperature adaptation, right? So and the term hologenome is coming most and most popular, no? So, and for you guys, I mean, you are coral researchers, so and probably the symbiosome is one of the most studied, symbiotic relationships studied in the world, right? But still, there are some, and especially all the related with the prokaryotic, associated microbes is not really well known still, the function they have, and some of them are even cryptic, so there's a lot of work to do still on this field. At the other level, I mean, and going back to the sponges here, I mean, we saw also that these make us to think into the concept of nested ecosystem. So, meaning that, all the relationships between the microbial partners and the host in front, and the capacity of reacting in front of environmental stresses depend on this interaction. So in this sense, it can end as a different scenarios, so stability or disease or even adaptation. So all what's happening in this holobiome will affect by a cascading effects first to the community and then to the ecosystem. So this is what, it reminds me all this type of connectivity, not just inside the holobiome, but also at the different levels of organization. So I will explain you some, examples on how this make us to, reorientate a little bit our research. So how looking at the organisms as holobionts, I mean, what was the consequences of that? One of these outcomes was the use, sorry. No, it's not. I don't know why it got, I know, I'm going, sorry, yeah, that's right, sorry. One of the outcomes is the use of dissolved organic matter as a food source. That was, a big finding, I think, and it's not that long ago now, and now we know that the sponges this is the removal, I mean, the delta of the DOC, meaning the difference between the inhaled DOC and the exhaled DOC in a sponge. So what we saw was like this removal is proportional to the DOC concentration in the water column. And this is happening because at the beginning we thought that this was just because the heterotrophic microbes associated to the sponges. But it turns out that, both strategies, these two sponges has two main strategies in relation to the amount and diversity of associated microbes. We call them either high microbial abundance species, when they have high densities and high diversity, or low microbial abundance species. It turns out that both strategies for the Mediterranean species, these are for the ones at the crambe, this is the Abra, Achelas oroides, Chondrosia reniformes, and Petrosia fififormis. So, they all follow the same pattern. So this was like, okay, this is not only bacteria, right, or microbes. So then, by a work led by Marta Ribes, we were able to find out, to demonstrate with two different Mediterranean species, a high microbial abundance one, which is Aplyssina aerophoba, and Decidea avara as low microbial abundance. We were able to demonstrate that for both of them, more than the 85% of the carbon source was due to the DOC. And not only that, but in the case of the high microbial abundant species, the main responsible was that 60% of this removal was because the sandbiome. But in the case of the low microbial abundance species, was mainly the sponge cells that responsible of this removal. And this, I can say that this, as much as we go in detail of this process, I mean, it is increasing the role of the host in this removal of DOC. And another, related with this, I mean, then was able to characterize other, biological pathways in terms of recycling the material in coral reef, for example. So because the ability of this is known as a sponge loop, and was discovered by the Guich and collaborators, and it is mainly that the sponge's capability of using this dome and transforming this dome to particles by means of fecal pellets, they can enter then to the tritivorous food webs. So now they are like, they're increasing the recycling of this material inside the coral reef. Right now, we are working on our current project. What we are trying to do is to evaluate the role of Mediterranean species transforming the dissolved organic matter, in the sense that we are wondering if they are removing the more labile DOM and they excreting, more recalcitrant DOM. So then they will act as a carbon pump in a similar way that the biological carbon pump or the microbial carbon pump. To do that, what we do is, these fluxes measurements and of a bunch of different compounds, and considering the two strategies, but also considering two different communities, a macroalgae community and a coralligenous community, and then also the seasonality. I'm very happy to see that, this role of the DOM is being evaluated in other groups of these ecosystems, because I'm sure, it will help for sure to understand the functioning, the overall functioning of this ecosystem. Another outcome of this, of looking at the organisms as holobionts, is the increase of the nutritional strategies. The typical, trait that actually we are, like describing being heterotroph of autotroph, is not very informative. But in fact, this is a simplification. As we know, this just means the carbon source is either organic matter or CO2, but also, the energy source or the electron donor play a role, right? So basically, photosynthesis are using light as an energy source, carbon as CO2 as a carbon source, and as an electron donor H2O2, and that's it. But when we consider the sponges, in this case, as holobionts, this turns out to this. So a bunch of different, possibilities appear, and all of those are already being found in either the enzyme or the gene associated to this enzyme. So, and most of them are waiting to be measured, in fact. So this, as you can see, open, a huge window of new possibilities, new interconnections, even not only inside the holobiont, not only at the community, but also these asterisks mean that those compounds are volatile, so they can escape to the atmosphere. So it is also, a connection with the atmosphere. I will talk a little bit about nitrification, which is probably the most well-known. And nitrification is just, the conversion of ammonia first to nitrate and then to nitrate, and this is only, it's only possible with microbes. So, and then this is connected to other pathways, and to which, they can generate gas compounds, and again, they can escape to the atmosphere. And for example, in this case, if there is, if this escapes to the atmosphere, is like helping a little bit the eutrophization, because it helps to remove nitrogen from the system. So all the components, all the enzymes associated to all these pathways have been described in sponges. All of them. So meaning that the potentiality of these, different processes is huge. In fact, we have now a lot of individual nitrification rates reports for many species around the world. And what we wanted to do is to see if in the case of the Mediterranean, we were able to see any signal of this nitrification in the water column. So to this end, what we did was like a cross-transect, we made cross-transect analysis at different distances of the wall, of a vertical wall, and we analyze nitrate, right? So what we found, this is the percentage of nitrate, near the wall, and this is the respect to, in relation to the farther point of the wall. So meaning that here, we did this in 2 consecutive summers, summer 21 and summer 22, and we did transects at 2 different depths, at 10 depths, and 10 meters and 20 meters, to see the difference between, macroalgae dominant community and the coralligenous. So what we saw was a clear accumulation now in the water column of nitrate. Without, and this is mostly clear in, for the deeper community. So this is, opens all these potential, different pathways that may happen in this near the bottom, or near the wall. And this is not only nitrogen. It has been also reported like a full pathway, some of them, for other compounds, such as sulfur cycle, and also for the methane, through the methanogenesis. So these, and also, the fluxes are not being measured yet. It's not easy, but they are not being measured. And some of them, again, are volatile, such DMS, or the precursor. DMSP is not volatile, but is the precursor of DMS. So, and also methane, which is an important greenhouse gas, right? So, volatiles, going back to volatiles, is the other outcome of this looking at the organisms as all of ions. Volatile organic compounds are considered as, are inside the, the black box of the dissolved organic matter, with the difference that these compounds can escape to the atmosphere. It's now to have, a lot of influence on the climate, because they are the precursors of the aerosols they formate aerosols, which are the precursors of the nuclei for the cloud condensation, the cloud formation, so modifying the climate. And also they can destroy the ozone, or can modify the lifetime of strong greenhouse gases, as such methane. From the biological point of view, what we know now, we just know that these volatile organic compounds are mainly produced by photoautotrophic organisms, being macroalgae, being plants, or being phytoplankton, right? But we know that they have a lot of functions. They have, they act as infochemicals, they act as defense mechanisms and combat oxidative stress, gas stress, light stress, thermal stress. They also protect from hydrostatic pressure and osmolyte and sulfur cycle and so on. So what we, and in fact, a recent PhD in my institute by Marta Ribes and advised by our colleague Rafael Simon showed that for a coral reef in Morea, the coral reef was a net producer, of most of the volatiles that were studied there. So if you compare with the offshore oceans. So what we wanted to do is to see if, because we found all the enzymes and all the genes, related to the enzymes in sponges, we wanted to know if Mediterranean sponges were, just using these compounds as for their metabolism. And we found, in this preliminary study with 3 species, this is the Avara, which is a low microbial abundance species, and 2 high microbial abundance, Aplysinairophora and Agelazoroides. What we found was, when comparing, this is inhaled water and this, the red, is exhaled water, when we saw that both high microbial abundance species were significantly removing polybromomethanes and also DMS, and also the precursor, DMSP, So, showing again, and I'm sorry, and also what we find out, and because the relationship between the cycle of the nitrogen and the sulfur, we wanted to know if the nitrifiers, which are the ones, the microbial that do the microbes that do the nitrification, were in somehow participating in this volatile metabolism. And what we did was at the lab was we just blocked the nitrification by using nitropyrin. So you add to your experiments nitropyrin, you block the nitrification, and automatically this removal of these volatiles stop. And when we stop, adding nitropyrin, the removal of volatiles starting again. So this is a nice, example of the co-metabolism inside the olive ion. So, it complicates the history, but still it's super cool. So we just now started a new PhD trying to, know more about all these processes. So all of these, examples is just a bunch of things, fluxes that remind us, the connectivity, not only inside the hollow ion, but between, the different species in the same community and also between communities. For example, in our case, in the Mediterranean, we are very interested to know how, all things produced by the macroalgae can be used, can be processed by the sponges and vice versa. So to do this and to understand it seems impossible not to talk about hydrodynamics. Hydrodynamics means so many things. Means transport, means dispersion, means filling up empty boundary layers, means cleaning these boundary layers, means a lot. And I think it should be included in this, approximation to the ecosystem studies, including them as a trait, in fact. So we tried to do that, and I must say it's not easy and it's not straightforward. So what we did was to, by means of this cross-transect approximation, we did, a different distance. This is a Mediterranean wall, vertical wall. We did at different distances from the wall. We measured a bunch of compounds, many of them, at two different depths, at 10 and at 20 meters. What we wanted to do is to relate, somehow, don't pay attention to the formula. This is an engineer approximation. I don't either understand very much. So, it's just, the message is that there is like a component which represent the hydrodynamics, the turbulent mixing coefficient. So, and we measure that by a bunch of acoustic Doppler velocimeter and acoustic Doppler profilers, so characterizing all the, the benthic boundary layer. And at the same time, you need that the spatial gradient of concentration, in this case, the nitrate, right? So we did it with this system. But what's the problem? The problem, and with this formulation, you can calculate the fluxes from the wall and towards the wall, right? And in fact, the idea was, because we have so many information about these communities, we know each species, what they are removing, the pumping rates, the size distribution, the relation between oscular size and pumping. So we were so ready to do these calculations and the study fluxes, just with the plain individual studies, but also compare these fluxes with these engineering approximations. But what was the problem? This engineering approximation was assuming that the velocity profile near the bottom was a log profile. So it was accomplishing what is called the law of the wall. But surprise, this works, in our flume studies, in horizontal, coral reef population, this type of things, or seagrasses, this type of things, but not in this vertical wall. So only 20% of the profiles were accomplishing the law of the wall. So wow, we are in a touch. So we need to look for other approximation. Probably, the eddy covariance methodology or something like that. But we are doing all this work with the help of Stephen Monismith from Stanford. Also, another related to the hydrodynamics, another thing is it complicates the history. For example, in summer in the Mediterranean, when we have this is depth, and this is the colors are water temperature, we can see here that there is like this strong stratification that some years, at 21, was the thermocline was at 40 meters. But the next year, was at 20 meters, and this change all the time. And this generates what is known as internal waves with a periodicity of 30 hours or 18 hours, that it really has a role in this, on this hydrodynamic, on this mixing, deep communities with shallow communities, but we don't know still how to, parameterize this. And we are, on our way to integrate all this data. So finally, as a little bit of summary and as a take-home message, I will say that, benthic communities, all the different communities are the topic of study in this symposium, are complex and dynamic biogeochemical reactors. If we go back to Gaia theory, they will be as the vital organs, as lungs, as kidneys, as liver, no? So, and by studying these individual traits under realistic conditions, doing experimental validation when needed, and also this approximation of the holobiont and taking the hydrodynamics as a key functional factor, we are able to explain the role of the different species at the ecosystem scale processes. This is a complex, this is a slow procedure, but it fills up with deep knowledge. And I think that due to this complexity and this dynamicity, it's really, it looks like unlikely that we will be able to copy, to reproduce this complexity in a meaningful way. So this highlights the importance of conservation of these ecosystems if we want to keep all the goods that they provide us. Thank you very much for your attention.
There's no microphone. Okay, thank you very much for this wonderful insight into the functional ecology of benthic organisms. I see the room has significantly filled up. That's great. Questions? Are there questions? Hopefully. But we need a microphone. I think we need a microphone. It's coming. Good.
Yes, hi, this is Line Bull Mortensen from IMR in Norway. I found this extremely interesting, your presentation. And I have a very short question, because you're talking about Gaia. And what about Gaia? Yeah. And it's about emerging properties that you can only study at a certain level to understand. And you have brought us through a really interesting travel from the laboratory out into the field, and we end up with internal waves. So it's a question, do you think that perhaps also starting in the other end of the scale and looking at emerging properties on your reefs that could not be measured at the detail level would also be interesting?
The question was if we can start, this approximation, for example, at the community level, or what? I'm sorry.
Yes, and I don't know tropical reefs very well, but I'm just thinking about emerging properties is a phenomenon we know about that can only be studied at a higher level.
Yeah,
and you are talking about reefs with all this diversity, and perhaps also, I'm just saying, by approaching from the other side, from the whole system side, in addition to this laboratory approach, could give us a really interesting picture.
Sure. I'm totally convinced of that. You need to, because it's so inspiring when you make a zoom out and escape from your comfort zone, no? Which are your, in my case, mostly Mediterranean communities, because it gives you a new perspective. We need to, look for other approximation, other also methodologies, I will say, because not all the methodologies are good for everything. It's not one size fit all in terms of methodology of ecosystems. So it really, definitely, I would recommend that for sure.
There's one over there.
Thank you, Marta. I'm Ulisse Cardini from Stazione Zoologica. Wonderful talk. I was very interested in the part on volatiles, and you showed that graph with in-X fluxes and different volatiles, and you said you saw a decrease in DMS with the sponge, and I was wondering, you saw both DMSP and DMS, right? And the MSP can be used as a carbon source or a sulfur source, but what about DMS, and where is the link with nitrifiers? Maybe you can elaborate on that a little.
I would like to, but we just started two months ago a PhD with this topic in collaboration with Rafael Simó from my institute. And those were like the preliminary, experiments just before, starting the PhD. So we are sure that we will get some results. So we don't know yet, but what we are planning to do is to, our guess is that these sponges are using, the volatiles produced by the macroalgae. Then enters, in which season, in which state, the maximum production of the macroalgae or the degradation of the macroalgae. There is a lot of things also to look at there. But also, we are planning to incubate macroalgae in the lab, and then give this water to the sponges, and then check for the activation of the of the microbial activity of nitrifiers, just to be sure that this is happening, no? And about the relationship between two cycles, I still haven't read much about, but there is some connection, because we had the intuition, that this may be related, so it was not because, with remit. I mean, we found it in some place. But right now, it's so incipient, it's so new that I don't have the explanation. Hopefully, after 4 years, we will know more about any, suggestion of if you know more than me on that. Just we can talk and we can.
We should.
We'll be happy to.
Yeah, thank you.
Are you measuring volatile? We can talk later. Yeah, we can.
Hi Marta, great talk. Jasper de Goey. I was wondering, so the hydrodynamics, I think a lot of us are not dealing too much with it because it's very complicated, but it's so important. But I was wondering if and how you also include the fact that sponges and others are actively filter feeding. So they're not only depending on the water flow, but they actually create current.
Yeah, good question. Yeah, I think this is happening just at the boundary layer, right? All this hydrodynamics. And we were thinking at the first approach to just go at the larger scale, mainly to know if in fact these walls are source or sink of compounds. This is another scale, because I think this is important, because I suspect that, for example, for DOM, some of the components of the DOM are generated in the wall. It's not coming from rivers, it's not coming from rain. They are generating, and they are just cultivating their own DOM that you don't find it at 3 miles away from the wall, Is this scale the first? The other scale is more, this is even more complicated, to introduce this, fine scale. And it's nice, but I think I'm more interested in the global thing than this, more engineering stuff, So.
Thank you, Marta. Very inspiring talk, very, very great talk. And just a very general question. I'm Carlo Cerrano, from the Polytechnic University of Marche. And considering all the functioning that you are studying, and considering what is happening with climate change in the last 20 years, we are losing very quickly a lot of this function in the benthic community. And in your opinion, sometimes we realize the role of species or the role of things, the value of things when we lose them. And sometimes the losing of gorgonians, the losing of sponges in some area open some idea on the role they play in the community. Before to start to analyze in the lab the detailed functioning of the species, now we have the opportunity to realize which are the effects of the species in the community. So with all your knowledge on this, which is more or less your expectation for how benthic community will change in the future, because we are losing a lot of these key species in the term of functioning.
This is a complicated question, Carlo. And because there are so many different processes, and even in terms, I can say, okay, if we lose high microbial abundant species, the nitrification will go low, and then, then the eutrophication will increase, this type of things, Or even, but the problem is also the hydrodynamics is showing a pattern due to the global warming. And we don't have other data of that. We saw, at least in our area, that the wind regime is changing. And this will modify. So then probably should go through modeling, which I'm not doing. I'm generating the basal data, but this should be because we don't have time to have all the possible conditions to say, this is what's gonna happen. No, because everything is going too fast. I think this is my opinion. But definitely for certain things, as when you lose, for example, Eunicella in the shallower communities, I mean, these sponges, phototrophic sponges living up there, probably they will suffer, because they hydrodynamics, for example. But yeah, other than that, it's difficult to say, yeah.
I don't see any questions anymore. Oh, there's one. Yeah, over there in the back. Can you turn it on?
Yes, yes. Thanks, Marta. Pedro Frade from Vienna. Just to try to see things from the smaller end of the processes. What I missed a little bit, and maybe you can tell a bit about that, is a lot of these processes, they cannot always happen in the same space and time, right? So there has to be some heterogeneity within these holobionts. And what is the next step in terms of looking at inside these sponges and cell-to-cell relationships and which microbes are doing which in relation to where they are positioned, right? And what is happening in that scope? Because I think that's also very important, because looking just at the holobiont as one black box can also be too generalistic, let's say.
Definitely. I mean, and we are, getting so good on observing these small scale things at the level of cell, at the level of organs, I mean, this is important, but it's another level. I'm an ecologist, I mean, big stuff. So I'm not gonna do that, but I think this is very important to link this acknowledge, this in detail acknowledge, because we are learning a lot of things. Yesterday we were talking with Jasper de Guy about the removal of VOM by pinocytosis, and knowing all these details, I mean, I recommend his talk, so to learn more about that, and is teaching us so many things, and even the ones that we like to measure the big stuff, the net result of the holobiont. I mean, it opens our mind to other possibilities, and this is fantastic, So definitely, I don't know where we should go, because it's not my field of, expertise. What kind of things should we, but there are many of them, so let them decide. But for sure, I will use this knowledge for, for open my, my looking at the ecosystem, my perspective for the ecosystems. And of course, I used to use all the new technologies available, collaborating with people that are specialists on this. So yeah, definitely. It's a great way of, keep learning, yeah.
Good, excellent, thanks. Massimo just reminded me that we should keep an eye on the watch. If there are no urgent questions, I think we should conclude this session. And the next sessions start at 11 o'clock sharp, I think. Now we have coffee break, so enjoy your coffee, and thanks again, Marta.